In 2016, a Japanese cell biologist named Yoshinori Osumi received the Nobel Prize in Physiology or Medicine, not for discovering a new drug, not for developing a new surgical technique, for describing a biological process that every single cell in your body runs continuously, a process that your cells have been capable of since before you were born, that declines in efficiency as you age, and that when properly activated, cleans, repairs, and renews cellular components in ways that have been directly linked to extended lifespan, reduced cancer incidence, lower Alzheimer's risk, improved cardiovascular function, and the reversal of measurable biological aging markers. The process is called autophagy, and the switch that turns it on is not a drug, not a supplement, and not something you need a prescription to access. It is a biological mechanism that your body already contains, and that specific identifiable daily behaviors either activate or suppress. Most seniors have never heard of AMPK. They have never heard of mTOR. They have heard of autophagy only in the context of intermittent fasting articles they may have skimmed and forgotten. What I want to do today is give you the complete mechanistic picture, how these systems work, how they interact with each other, what suppresses them in the daily habits of most older adults, and what the research shows about activating them in ways that are measurable, reproducible, and accessible today, not eventually. Today. I am Dr. Edward. What you are about to hear is educational information based on published scientific research, including Nobel Prize-winning biology. It is not a substitute for personalized medical care. If you have diabetes, cancer, or are currently undergoing any treatment protocol, the interventions I describe may interact with your condition or treatment and must be discussed with your physician before implementation. Subscribe to this channel right now. This video covers the most fundamental biology of aging that I have ever addressed on this channel, the cellular machinery that determines how fast you age at the molecular level. It is free, it takes 2 seconds, and this is the kind of research that changes not just what you do, but why you do it. Subscribe and stay with me. Let me start by giving you the framework, the 3-system architecture of what I am calling the longevity switch, because these 3 systems work together and you cannot understand any one of them without understanding how they interact. The first system is AMPK, adenosine monophosphate activated protein kinase. AMPK is the cell's primary energy sensor. It monitors the ratio of AMP to ATP, energy-depleted molecules to energy-available molecules, and when that ratio shifts toward depletion, AMPK activates. Think of AMPK as the cell's fuel gauge. When the needle drops below a certain threshold, AMPK turns on and initiates a cascade of responses designed to conserve energy, generate more of it, and shift the cell from consumption mode into maintenance mode. AMPK activation triggers mitochondrial biogenesis, the creation of new mitochondria. It promotes fatty acid oxidation, the burning of stored fat for fuel. It activates autophagy, the cellular recycling program, and critically, it inhibits mTOR, the growth and construction program that, when chronically active, is one of the primary drivers of accelerated aging. The second system is mTOR, mechanistic target of rapamycin. mTOR is essentially the opposite of AMPK. It is activated by nutrient abundance, by growth signals, by insulin and IGF-1, and by amino acid availability. When mTOR is active, the cell is in anabolic mode, building proteins, expanding its components, preparing to divide. This is appropriate and necessary in periods of growth, recovery from acute stress, and tissue repair following injury. The problem arises when mTOR is chronically active, when the cellular environment never signals scarcity, never removes the foot from the accelerator, never allows the maintenance programs to run. Chronically elevated mTOR suppresses autophagy, promotes cellular senescence, the state in which damaged cells stop dividing but refuse to die, and instead release inflammatory signals that damage surrounding tissue, and accelerates the accumulation of misfolded proteins and dysfunctional organelles that characterize aged tissue. The third system is autophagy itself, literally self-eating, from the Greek. Autophagy is the cellular process by which cells identify their own damaged, misfolded, or dysfunctional components. Proteins that have been oxidized or aggregated, mitochondria that are producing excessive reactive oxygen species, entire sections of the endoplasmic reticulum that have accumulated unfolded protein stress, and package them into double-membrane vesicles called autophagosomes, which then fuse with lysosomes containing digestive enzymes. The damaged components are broken down, and their molecular building blocks, amino acids, fatty acids, sugars, are recycled for use in new synthesis. Autophagy is, in the most literal sense, the cellular cleaning and recycling program. When it runs efficiently, the cell maintains a high quality of its internal components. When it is suppressed, as it is by chronic mTOR activation, by chronic caloric surplus, and by several of the daily habits I described in my previous videos, damaged components accumulate, cellular function declines, and the tissue-level manifestations we call aging accelerate. Research published in Nature Cell Biology established that autophagy declines significantly with age in multiple tissue types, and that this decline precedes and predicts the development of age-associated diseases, including neurodegeneration, cardiovascular disease, metabolic syndrome, and cancer. Autophagy is not one of many mechanisms of aging. It is one of the most central, and its restoration in aged organisms, through the specific interventions I am going to describe, has been shown to extend lifespan in every model organism in which it has been tested, from yeast to roundworms to fruit flies to mice. Subscribe to this channel right now if the architecture I just described is making sense and you want the practical translation. Every single week, I take this level of research and turn it into something specific that changes what you actually do. Subscribe. It is free. And let us go deeper. Now let me explain what activates AMPK and autophagy, because this is where the research becomes directly actionable. AMPK is activated by anything that creates a transient controlled energy deficit in the cell, a state in which the cellular energy demand temporarily exceeds the immediate supply, shifting the AMP to ATP ratio toward depletion. There are 4 specific ways to create this state that have the strongest evidence base and the most direct relevance for seniors. The first is the extended overnight fast that I described in a previous video. When you stop eating for 12 to 16 hours, hepatic glycogen, the stored glucose in the liver, is depleted. Blood glucose and insulin fall to baseline. The AMP to ATP ratio in liver and muscle cells begins to shift. AMPK activates. mTOR is inhibited. Autophagy initiates. Research from the laboratory of David Sabatini at MIT, published in Science, mapped the precise time course of autophagy induction during fasting in mouse tissue and found measurable autophagosome formation beginning at 12 hours of fasting and peaking between 16 and 24 hours, returning to baseline with refeeding. The 12 to 16 hour window is the practical sweet spot for most seniors, long enough to induce meaningful autophagy without creating the physiological stress of more extended fasting. What this means practically is the same recommendation I gave before, but with the molecular mechanism now fully explained. Finish eating by 7 or 8 p.m. and do not eat again until 7 or 8 a.m. This is not a radical dietary restriction. It is a restoration of the daily eating pattern that human physiology evolved alongside, one in which the nightly fast was a natural consequence of the absence of artificial lighting, refrigeration, and 24-hour food availability. The ancestral fasting window was not a diet. It was simply the rhythm of a day that ended when the light did. The second AMPK activator is exercise. Specifically, the type of exercise that creates a meaningful but sustainable energy demand in skeletal muscle. When muscle cells are contracting against resistance or at moderate aerobic intensity, their ATP consumption rate temporarily outpaces their ATP production rate, activating AMPK through the energy deficit I described. AMPK-activated muscle cells upregulate glucose uptake through insulin-independent GLUT4 translocation, increase mitochondrial biogenesis, and initiate autophagy in the contracting muscle fibers, clearing the damaged proteins and dysfunctional mitochondria that accumulate with age and with the mechanical stress of exercise itself. Research from the journal Cell Metabolism found that a single session of moderate-intensity aerobic exercise produced measurable AMPK activation and autophagy induction in human skeletal muscle biopsies, with the magnitude of autophagy proportional to the intensity and duration of the exercise session. More importantly, a separate study in the same journal found that exercise-induced autophagy was essential for the metabolic benefits of exercise. When autophagy was genetically blocked in mouse models, the improvement in insulin sensitivity and metabolic efficiency from exercise was abolished. The muscle was working. The cellular cleaning program that makes the work beneficial was not. Autophagy is not a side effect of exercise. It is a primary mechanism through which exercise produces its benefits. The practical prescription is 30 minutes of moderate-intensity aerobic activity, brisk walking, cycling, swimming, 5 days per week, combined with 2 to 3 sessions of resistance training. The aerobic component drives cardiovascular AMPK activation and systemic autophagy. The resistance component drives skeletal muscle AMPK activation, preserves the muscle mass that is the primary site of glucose disposal, and prevents the sarcopenia that is one of the most powerful accelerators of metabolic aging. The third AMPK activator is thermal stress, specifically brief exposure to cold or heat that creates a controlled physiological challenge. Cold exposure activates AMPK through brown adipose tissue thermogenesis, which has an extremely high energy demand and produces rapid AMP accumulation. A study published in Cell Reports found that brief cold water exposure, 3 minutes at 14 degrees Celsius, produced measurable AMPK activation and autophagy induction in circulating immune cells, with effects persisting for several hours after the cold exposure ended. Sauna exposure activates AMPK through heat shock protein pathways and has been associated with dramatically reduced cardiovascular and all-cause mortality in the Finnish epidemiological literature. A Finnish cohort study published in JAMA Internal Medicine found that 4 to 7 sauna sessions per week was associated with a 40% reduction in all-cause mortality over a 20-year follow-up. The mechanism involves both AMPK activation and the direct cardioprotective effects of heat shock proteins. I am not prescribing cold plunges or daily saunas for every senior watching this video. I am pointing out that the physiological mechanism underlying these practices, controlled thermal stress producing AMPK activation, is real, documented, and accessible at various intensities. A brief cold shower in the morning, a hot bath in the evening. These are not trivial wellness practices. They are specific AMPK stimuli that activate the longevity switch at a modest but cumulative level. The fourth AMPK activator, and perhaps the most underappreciated, is phytonutrient activation through specific plant compounds that directly interact with AMPK signaling pathways. Several naturally occurring plant compounds have been shown to activate AMPK through mechanisms that partially mimic the energy deficit signal without requiring actual caloric restriction. The most extensively studied is EGCG, epigallocatechin gallate, the primary catechin in green tea. A meta-analysis published in the journal Nutrients analyzing 21 randomized controlled trials found that EGCG supplementation significantly activated AMPK in human subjects, with downstream effects on adipogenesis, glucose metabolism, and inflammatory marker reduction. Green tea's cellular mechanism of action is not antioxidant capacity. It is AMPK activation. Resveratrol, found in red grapes, dark berries, and Japanese knotweed, activates SIRT1, the primary longevity sirtuin, through a mechanism that is partly AMPK dependent and partly direct. The human evidence for resveratrol is more contested than the animal model evidence, with bioavailability being the primary limiting factor, but the mechanistic case for resveratrol as a longevity switch activator is strong. Berberine, a compound found in golden seal and barberry, is perhaps the most potent AMPK activator among plant compounds, so potent that multiple meta-analyses have found it produces blood glucose and lipid improvements comparable to metformin through the same AMPK mechanism. And quercetin, found in onions, apples, and capers, activates both AMPK and autophagy and has specific evidence for clearing senescent cells, the zombified aged cells that accumulate and drive inflammaging. Now I want to connect all of this back to the aging biology because I want you to understand not just the how, but the why. why activating these systems with such consistency produces the life-extending effects that the research documents. When AMPK is chronically activated and mTOR is appropriately inhibited, which occurs during the nightly fasting window, during exercise, and during the physiological stress of thermal and phytonutrient stimulation, several things happen simultaneously in the cell. Autophagy clears the damaged proteins that would otherwise form the aggregates associated with neurodegeneration, the oxidized lipids that damage arterial walls, and the dysfunctional mitochondria that produce excessive reactive oxygen species and trigger apoptosis. Mitochondrial biogenesis replaces the degraded mitochondria with new, functional ones, restoring cellular energy production efficiency. Cellular senescence is reduced because the accumulation of damaged components that triggers the senescence program is interrupted by their autophagy-mediated removal. Telomere attrition slows because the oxidative stress that damages telomeric DNA is reduced when mitochondrial quality is maintained, and the systemic inflammation that underlies most age-related disease produced by senescent cells, by circulating damaged proteins, and by the innate immune response to cellular debris falls as the cellular quality that generates those signals is restored. Research published in Science Translational Medicine examined the effect of restoring autophagy in aged mice through genetic and pharmacological means on multiple aging biomarkers simultaneously. Restored autophagy was associated with measurably longer median lifespan, reduced senescent cell burden, improved cardiac function, reduced neuroinflammation, and improved performance on cognitive and physical functional tests. The authors concluded that autophagy decline is not a passive consequence of aging, but an active mediator of it, and that its restoration is one of the most potent single interventions available in experimental gerontology. I also want to address something that I think most seniors find difficult to integrate with what they have been told about nutrition for the past several decades. The advice to eat frequently, 6 small meals a day, never skip breakfast, always have a snack between meals to maintain blood sugar, was developed in a context of managing acute metabolic symptoms rather than optimizing long-term cellular aging. It is advice designed to prevent the immediate discomfort of blood sugar swings. It is not advice that was ever evaluated for its effect on AMPK activation, mTOR inhibition, or autophagy. When those outcomes are measured, as they now have been in multiple studies, frequent eating is one of the most consistent suppressors of the cellular maintenance programs that determine biological aging rate. This does not mean seniors should be chronically undereating or that protein intake should be restricted. It means the timing, pattern, and composition of eating matters as much as the total quantity, and that the nightly fasting window is the most important single dietary variable for cellular aging that most seniors have never been told to protect. Subscribe to this channel right now if this is giving you the framework to understand why the specific interventions I recommend across all of my videos connect to the same underlying biology: the fast, the exercise, the dark bedroom, the stress recovery window, the phytonutrients. All roads lead to AMPK activation, mTOR inhibition, and autophagy. Subscribe, it is free, and stay with me for the practical protocols. Here is the complete activation protocol, because I want to be as specific as possible about what to actually do. Morning. Do not eat immediately upon waking. Allow the overnight fast to extend as long as is practical and comfortable, ideally to at least 12 hours from your last meal the previous evening. During this pre-breakfast window, drink green tea. A single cup of properly brewed green tea delivers approximately 100 to 150 milligrams of EGCG, a meaningful AMPK stimulus that extends the metabolic benefit of the overnight fast. If you exercise, doing so in a fasted or semi-fasted state before breakfast or 2 to 3 hours after a light breakfast maximizes AMPK activation during the session because the energy deficit signal is stronger when glycogen stores are partially depleted. Midday. Make lunch your largest meal. This is the time in the circadian cycle when insulin sensitivity is highest, when nutrient partitioning is most efficient, and when mTOR activation from dietary protein and carbohydrate is most rapidly resolved. The same meal eaten at noon produces less sustained mTOR activation than the same meal eaten at 9 p.m. because circadian insulin sensitivity is lower in the evening and the metabolic processing is slower. Evening, aim to finish eating by 7 or 8 p.m. The earlier this window closes, the longer the overnight fasting window extends, the deeper the AMPK activation during sleep, and the more complete the autophagy cycle before the morning meal. Include quercetin-rich foods, onions, apples, broccoli, in your evening meal. The quercetin activation of autophagy compounds with the fasting-induced AMPK signal. Twice weekly, include a session of meaningful physical exertion, not a casual walk, though casual walks have value. A session that creates genuine cardiovascular effort or muscular fatigue. The energy deficit created by real physical effort is the most potent AMPK stimulus available outside of pharmacology. Daily, 10 minutes of slow diaphragmatic breathing or a 15-minute walk in a natural environment. Both reduce cortisol, reduce PARP1 activation, and protect the NAD+ pool that the sirtuin system, the downstream partner of AMPK, requires to function. Here is the summary of everything we covered. The longevity switch is not a metaphor. It is a specific molecular system consisting of AMPK, mTOR, and autophagy that determines the rate at which cellular maintenance runs versus cellular aging accumulates. AMPK activation turns the switch on. mTOR inhibition allows it to stay on. Autophagy is the maintenance program that runs when the switch is on. AMPK is activated by the energy deficit of the extended overnight fast, by moderate to vigorous exercise, by controlled thermal stress, and by specific phytonutrients including EGCG in green tea, resveratrol, berberine, and quercetin. mTOR is chronically suppressed by the same fasting window that activates AMPK. Autophagy runs during the periods of AMPK activation and mTOR inhibition, primarily overnight and during exercise, clearing damaged proteins, replacing dysfunctional mitochondria, reducing senescent cell burden, and interrupting the inflammatory cascades that drive age-related disease. The daily protocol that activates this system is a 12-hour overnight fast, green tea in the fasted morning window, exercise in a semi-fasted state twice weekly, quercetin-rich evening meals, and a daily physiological stress recovery window. Please share this video. Share it with the person in your life who eats 6 small meals a day because their doctor told them it was good for their metabolism. Share it with the senior who has never understood why intermittent fasting shows the results it does. Share it with anyone who wants to understand not just what to do, but why, at the molecular level, it works. The longevity switch is on in every cell of your body. These are the behaviors that activate it. Subscribe to this channel if you have not already. My next video covers the specific supplements with the strongest human evidence for directly supporting AMPK and autophagy, the compounds that extend the effect of the protocol I described today into the hours when fasting, exercise, and phytonutrients are not doing the work. Subscribe so it comes directly to you. I am Dr. Edward. The switch is inside you. Turn it on. I will see you in the next one.